The overall goal of the following experiment is to rapidly obtain amyloid fibrils from any protein in vitro. This is achieved by dissolving the protein of choice in MES buffer to induce structure misfolding. The protein can then be incubated at high temperature, which promotes the formation of amyloid in the absence of other cofactors. Alternatively, the protein is cross-linked by EDC in order to generate stabilized soluble protein oligomers, the precursor of amyloid. Finally, non-protein cofactors are mixed with stabilized soluble protein oligomers to allow the formation of hybrid amyloid. Results are obtained that show native proteins can be readily converted into amyloid conformation when facilitated by favorable conditions.
This method can help answer key questions in protein misfolding diseases, such as how precisely amyloid forms from soluble protein oligomers, and whether different types of amyloid have distinct biological and pathological functions.
To begin, prepare MES buffer as described in the text protocol for both protein-only amyloid and stabilized soluble protein oligomers. Then, weigh the protein of choice and reconstitute it to 10 mg per milliliter in MES buffer. In this demonstration, human serum albumin, or HSA, is used. Incubate the solution at 65 degrees Celsius in a water bath for 4 hours. By the end of the period, a white precipitate is visible in the solution. Neutralize the sample with 10% volume per volume tris HCl. Dialyze the protein in a dialysis cassette overnight to exchange to the desired buffer for further analysis. Prepare a control sample of native protein by following these steps without